Alright, the first step in every lab is to always make sure you're following proper safety rules, which means, in this case, proper PPE like gloves and safety glasses. And we're going to start by making our gel by taking about 2 grams of the agarose and adding it into a beaker, and then about 100 milliliters of the TAE buffers shown here. Mixing it up in a beaker here and then microwaving in total for about 1:30, but you want to make sure it doesn't boil. So be careful of that, and it will be hot when you pull it out of the microwave. And the next thing you want to do is start by prepping your gel tray. So you want to make sure you tape up all those sides. Leave the comb off for now, but make sure you tape up all those sides to not let anything seep through and out of it when you're pouring your gel in later. And since I don't have an active sample for the day, we're just going to look at this gel tray for a little bit. But we're adding in 2 microliters of ethylene bromide into the mixture that we had in our previous picture. You want to pour about 3/4 of that into the gel tray. Then add in that comb. You want to make sure you add it in at the end and not the middle. And then you can finish pouring the gel, making sure that the comb is set in there correctly. And while we are waiting that 15 minutes for it to cool down, you are going to need to prep your samples to put into the wells in your gel. And for this electrophoresis, you need about 3 microliters of loading dye per well. And if you are using a DNA ladder, you do need 3 microliters of that. And then depends on how many DNA samples you have. That depends on how many wells you need to prep for the loading dye. But in this case, we only had 2, so we only needed 3 microliters of my sample and 3 microliters of Elma's sample, and 3 microliters of loading dye for each of those samples. Okay, now it's time for the good part, setting up that actual electrophoresis rig. Make sure you fill that up with TAE buffer up to the ledge, and then when your gel is cooled, go ahead and place that in with the wells facing the black voltage. Then continue filling that TAE buffer just so it's slightly over the top of that, because you don't want too much liquid over the top of the gel, but just enough. And then you'll want to put the lid on and close it up, making sure that the voltages run to the right colors, red to red, black to black. Make sure it's plugged into the electrophoresis rig and turn it on, and you're going to run that for about 30 minutes at 100 volts. But I do believe we modified it for a different week, so just check with your instructor on what you need to do there. And then at the end, you will take gel imaging of it. So after that 30-minute wait, you'll go ahead and take it out, put it in the gel imager, and you will get a result.